So in terms of the focus for the panel this morning, the focus will be discussing the concept of platform validation, specifically focusing on genome editing. And as an organization, Clearview has worked extensively within the gene therapy space, and we're exceptionally excited with the milestones that we've achieved over the course of the last year or two. And with the concept of today, what we want to focus on is recognizing that when we're starting to think about genome editing and both the promise as well as the expectation of what does it mean to permanently edit our genetic code, what we want to open the discussion about is how do we think about both validating platforms and how do we build trust as well as confidence with regards to genome editing, and how do we think about what are the different stages of platform validation, as well as what does it mean to think about platform validation. And our goal is to spend time thinking about that, as well as where are we going from here? What is the potential of genome editing when we think about 15, 20 years from now? How do we build towards the potential vision of delivering cures for thousands of indications that today are not being addressed? So I'm actually going to let the panelists introduce themselves. So it'd be great if you could introduce yourselves as well as provide just brief context with regards to your given platforms.
So Katrine Bosley, I'm the CEO of Editas Medicine, and believe it or not, we are not yet 4 years old. So I think it gives you a little bit of a sense of maybe the speed with which this field is moving, that we, as one of many companies in this field, have had the benefit of rapid progress. But I think also as we think about how do you establish a platform with a new technology, gene editing is not new, but CRISPR has relatively newly emerged. There's a tremendous lift to go from a cool science in the laboratory to actually making medicines. And I think one of the there's always there's lots of stuff in the media about it these days. You constantly read how cheap, fast, and easy it is. And I think everybody in this room knows it's not cheap, fast, and easy to make a medicine. There's a big difference between doing interesting science in a lab and making a medicine. That's why we're here. And in particular, to translate the science of CRISPR, both CRISPR-Cas9 and CRISPR-CPF1, into medicines, not only for genetically defined diseases, which is where I think we all start when we think about repairing broken genes, but also what we call genetically treatable diseases. I mean, they're when you understand the biology well enough, the world of potential therapies is not limited to those where it's actually mutation driving the disease. If you have sufficient understanding of the biology, editing a gene to therapeutic benefit makes it a genetically treatable disease in other situations. So we do both.
Thank you. Welcome. Matt?
Yep. So, excuse me. So again, Matt Kane, part of the team at Precision Biosciences, also the genome editing company. And first of all, thank you for having us here today. It's great to be even just a part of this panel. A lot of people I really respect here. Again, a genome editing company, 12 years old, not a CRISPR company. I have to make that clear. We've developed a genome editing platform that we call ARCUS. We spent really the last 10 years developing this platform, all with the mindset of can we actually edit for therapeutic benefit. So some of the key problems that we have attempted to solve with this particular platform, delivery, random off-targeting, and the ability to control the edit. We think we've got a good handle on these things, and you'll be hearing a lot more about this technology a little bit later this afternoon. I think our head of business development, Mike Dombeck, is going to be speaking around 1 o'clock, and encourage you to attend that. Thanks.
Sandy.
Hi, I'm Sandy McRae. I'm the CEO of Sangamo, and we're the senior partner here. We've been around for 22 years as Sangamo Biosciences and are now Sangamo Therapeutics, which was an important decision we made in June of last year, and it was relaunched at the beginning of this one, that we're not here just to do science. We're here to apply that groundbreaking science to important diseases and to patients' lives. And once you have that as your mindset, it focuses the whole company. Like Matt, we agree that there are important ways to think about gene editing, and we try and think about it as precision. Can you cut nucleotides within the genome, and can you get to all the places you want to get? We think about it as efficacy. Once you get there, how effectively do you cut at that site? And we think of it importantly about specificity. Are there places other than the place you want to cut where there may be off-target effects? And what we're learning as we go through this journey is the three of those are very tightly related. For many of the technologies, as you raise one, you unfortunately raise one of the others. So we're delighted to be part of this because I think validating the platforms sounds like an accountancy issue or a box checking issue. But we at Sangamo, with 4 trials open and about to dose the first patient for in vivo genome editing, it's a real responsibility that it hasn't just to be validated on a slide, but that we as a company have got to feel the certainty and the responsibility of about to give it to a patient.
Last but not least, Albert.
Yep. So first, thank you for inviting me to be on the panel. So Albert Seymour, I'm the CSO of a company that's not even 2 years old. We started in 2015, the end of the year 2015. And what we're doing is we're developing a technology using recombinant AAV vectors. We have 15 of these novel vectors, and really trying to exploit this new property that we found that's been described for more than 20 years, that AAV can induce homologous recombination-based gene correction. And this is something that we're now exploring and pushing our technology to address different genetic diseases. And so along with the other panel members, I think one of the ways that we look at validating this technology platform is taking and riding on the background and the mechanism of homologous recombination, this highly precise mechanism that cells naturally undergo anyway during cell division, to then apply that to diseases where we feel that there's significant unmet medical need, but during that validation, also aligning it with genetic diseases where we have some idea, based on human genetics, how much we need to correct in order to have a therapeutic benefit. And that's where this inflection or this intersection of human genetics is now coming to a position where these technology tools that we have access to can come together. And that's sort of one of the aspects that we focus on aligning those two and then validating around the idea around precision and specificity and safety and efficacy.
Great. So Sandy, I wanted to start with you. You mentioned, we're waiting for dosing the first in-human in vivo trial, and I wanted to understand, what does this mean to Sangamo, and also, how did you think about when, as an organization and as a platform, you were ready for that, that leap into the clinic, because it's such a big milestone that everyone has acknowledged, and everyone has acknowledged the responsibility of taking that step. I'm sure there were lots of internal debates. It'd be great to hear a little bit about what the dialogue was like internally.
So it means a great deal to the company. It means a great deal for how we are appreciated in the financial part of the company, of course, but it's much more than that. It's a sense of purpose that as CEOs we all have to inculcate in our people and encourage that there's a passion around doing something meaningful for patients and doing good for patients. There's something about scientists that have given the whole of their scientific career to work on zinc fingers, realizing that their dream has come true. That is a very beautiful thing, and a thing I feel a great responsibility for. But it isn't done just on a single decision. And it isn't done simply at the approval of either the FDA or the RAC. It has to be an internal company decision that you yourself feel the time is right to do this. And if you look through the various stages that Sangamo has done, with our zinc finger technology, we've been able to edit in vitro in cells for a long time. And if you look at our recent T-cell data, we can now do T-cell editing at 90, 95, even 99% efficacy. And then you need to look in animals, and we've got lots of mouse data where we can do in vivo genome editing in the liver, and now we even have some beautiful data where we can do in vivo gene transcription control in the brain. Then you need to move to large animals, and we have evidence that we can do this in monkeys. And it's surprisingly difficult, that transition between small animals and mice and monkeys. And so it's something that is a coming of age for all of the companies when you achieve that in large animals. And then there's something about your first dosing in humans. And it's less appreciated than it should be that there's 102 patients walking around already where Sangamo has edited their T cells. And they've had at least 3 years of follow-up. Their T cell reservoir has been reduced, their CD4 counts are up, the patients are safe and well, and the editing works and it lasts. So this last stage to do in vivo genome editing isn't as big a leap as it seems. It's a big leap emotionally and scientifically, but we can only do this because we've built a very strong foundation of scientific understanding and prudent steps to get there.
How about the choice around the first indication and the first program?
So we feel that at the moment we should balance medical need with, should be the first and foremost thing you use as your choice. So diseases which can be easily treated or don't have an important medical need are things that I hope eventually gene and genome editing will be able to address. I think the best example of our in vivo genome editing program is MPS 1 and 2, Hunter's and Hurler's syndrome. And our goal there is to get to children as early as possible and as young as possible, so as we can make that change in their life and give them a new copy of the gene that will prevent the changes that leads to them being in a wheelchair, their life being shorter than it should be, and all the sequelae. Mm-hmm. But to get there, we sometimes have to make prudent early steps. So we start in adults, and that is right because they can do an informed consent, but it's only a stage that then takes us to children, because I think that's where the benefit of genome editing is.
If I might just maybe add a comment on that, because I think for all of us, when you have a potentially broadly applicable platform, how do you pick your first indication is something you spend a lot of time on. Mm-hmm. What's technically tractable? Where's the clinical need? What's the developability, which is something that all modalities deal with, not just gene editing. But I think when you have a new modality, as we all do, you're also looking for something else, which is how can that first program is going to carry the weight of the platform, right? Both positive and negative. And the last thing you want is either false negative or false positive in your particularly early clinical readouts. And that first program should, in and of itself, be a good choice, but also open up the rest of the platform. And so, you're going to develop it in a way that you're going to apply in future programs, so you should get a lot of leverage out of it technically. Mm-hmm. And it also should help you reduce the risk of the rest of the platform. And so that's something that we think about at Editas, but it's actually my third platform company. And the other two, one was a biologics protein company and the other was a small molecule company. And it's the same question, frankly, regardless of the technical modality. But if you don't get that right, then particularly as you're a young company and you're building yourself up, it's hard to justify the cost of capital from the early stages of the company because it's very high cost of capital. And so you really need to wring as much as you can out of that early program above and beyond its importance as an individual medicine, which is, of course, paramount.
So maybe Albert and Matt, leaping off of what Katrine said, how do we think about what are some of the earliest questions or learnings we want to establish so that we're building from some of the earliest programs and earliest indications that we might be targeting?
Well, just to kind of build off what Katrine was saying, I think we absolutely agree. I mean, there are diseases like Duchenne's that we would love to apply our platform to today, but from a technical risk perspective, it's just simply too high for us right now. So it is absolutely critical that our first programs are successful, meaning that they're clearly bringing a positive benefit to the patient and that they're safe. And so that guides us down a very different path in terms of the sort of programs that we select. And I think by being successful in these earlier programs, it will open the door up for us to explore some areas that, frankly, have a greater potential impact for editing in general.
Yeah, and I think, along the same lines, I think identifying those indications where there is significant unmet need, but technically, from human genetics, you can address that need with the technology in hand, knowing that all of these are brand new, transformational, cutting-edge technologies. Go after those diseases where that unmet medical need is there that we can deliver to, and that there's some element of preclinical models that we can use to get that dose right. Because I think that's also something that's critical, that we come into these patients with your first dose. In our case with AAVs, we want to get that right the first time so that they will not be able to be redosed.
So you can actually have confidence
so
that
you
you're
want to
working.
have confidence that
right?
it's going to work.
You're not targeting an indication where you can't even measure, what is the clinical effect that we're having.
Exactly. And I think to follow on that too is just also establishing that path with the community and the regulators of trust, because I think we heard a little bit about ARM, that we're moving forward with these genome editing technologies. What are the assays that we're putting in place with that first indication that we're going in that we can reliably assess precision, on target, as well as dose response prior to going into those patients? I think that really helps build the platforms and help validate that as we progress in that first patient population.
Yeah, actually that's a great transition to another topic that we wanted to explore. During several of our early calls in terms of prepping for this panel, several of you mentioned, everyone is talking about specificity. Everyone's talking about the importance of specificity in building trust because there's a shared responsibility in terms of making sure that there's comfort with these programs, but that there is a lack of common nomenclature, common standards with regards to assays. And I am somewhat curious as to, what are some of the efforts that are ongoing? Are there shared conversations amongst yourselves? Are there avenues for collaboration so that we can make sure that there's continued forward progress for all of your programs?
Well, ultimately, this is a scientific question, right? And so part of our approach has always been, one, to present a lot. We've presented a lot on this, and not only in terms of there's really two parts to this problem. Can you optimize a molecule to be very specific, and what are the ways you can optimize it? There's a second part, though, which is how do you know? What are your analytical methodologies to do that? And there are some that are in the literature, and certainly we use those. We also felt that there wasn't sufficient robustness of some of those methodologies, so we created some new methodologies, which we've presented on as well. I think that it's funny because in the world of CRISPR, there was this early publication by one of our founders, Keith Jung, where he was testing a method that he developed to assess specificity, and he purposely used dirty molecules in that method because he was trying to test his method. And it's weird because that paper gets kind of referred to sometimes, and people are like, it's hard to make a specific CRISPR. He's like, Keith is always like, they missed the point. Right? He also has in that paper some very specific guides, but I think he was certainly, hey, all of us can make dirty molecules. We can make dirty small molecules. We can, we don't call them drugs, right? So we can make specific Cas9 or CPF molecules, CPF1 maybe more specific even. There are a lot of ways to optimize specificity, and then what's important, though, is to look by multiple orthogonal methodologies at specificity. In silico as a starting point, it's not sufficient, right? So you need to look in silico, you need to look biochemically, you need to look by cell-based methods. You need to look both in a targeted manner, where do you think you might have a sense that there might be higher risk because it's related to your on-target sequence, but you must look in an unbiased, comprehensive manner as well. And by looking robustly, that's how you get the confidence to know that you're not seeing any off-targets. And every target we've worked on, we find those molecules quite readily. That said, we also think you don't stop, right? Because I think there are very interesting ways to further engineer the molecule. In the case of Cas9 or CPF1, it's a two-part molecule. You have a protein portion and you have an RNA guide portion. Both are opportunities to drive specificity. There are ways you can actually create high-fidelity engineered variants of the protein that further enhance specificity. So there's a lot of tools in the toolkit to really tune up the molecule and get 90+% efficiency and no detectable off-targets by 6 different methods.
Sandy, you look like you were...
Yeah, I think Katrine's correct in that many of the medicines we give now have all drugs have had side effects. Paracelsus, all drugs are poisons and have side effects. And I used to chair the safety board of a large pharma company and have to decide if a drug was safe to be released into the community and think about adverse events. And it was easy, relatively, because if the patient took the drug and it didn't agree with them, the drug washed out their system in a day or a week and the event would pass. What we're doing is much more important, or much more has a greater sense of responsibility, because we're stitching in a permanent change to the genome that that patient will have with them for the rest of their days. And we, I think, have an obligation to follow them long term, to understand that so that we can better decide what the benefit risk is. We all, I think, use quite similar methods for looking for off-target. It should be standardized. I think when we go to the agency and talk with the FDA or with the RAC, they are increasingly asking us all to do similar things. We have the advantage that we're leading many of those discussions. And we just got another IND approved where we were pushing methods I don't think anyone else has tried yet to show that we can drop off-target and the consequences of target as low as possible. We're now at the limit of detection for off-target. But there must be more. We must keep striving to look for more. Because off-target effects, I believe, wouldn't be seen immediately you do the experiment. They may not even be seen a week or a month or a year later. If you truly are making cuts somewhere else in the genome that is deleterious or is affecting another gene, it may be 10, 20, 30 years down the road before those effects are seen. That's why we need to start with diseases where the benefit to the patient is sufficiently great. Chemotherapy has probably similar effects and pro-cancerous effects that is a more close balance and a more close analog to what we're doing.
Yeah. And just to follow up on that, I think the other thing, when you look across this panel as a whole, there are four of us here, each have very unique different technologies. So standardizing against something, there's cutting the DNA, there's a double-strand break, there's a single-strand break, or there's homologous recombination without really adding any endonucleases. And so pulling together some kind of standardization that perhaps is at the level of where we all convene, and that's at the DNA level, because I know that a lot of these assays where we will test for very unbiased off-target effects by looking at what did we put into the genome and not asking where we think it might go, but asking the genome, where is it, and going out and fishing that out, using some kind of standardized DNA line or something so that we're all comparing apples to apples, particularly when it goes to regulators or as we could collaborate with each other.
But the lesson from other medicines as they get approved is the first company to get approved or the first agent to get approved will most likely set the standard for how others are looked at.
So don't screw this up.
But I think it's interesting too. The FDA is very clear in this, not just with gene editing, but with all modalities, as they say, they don't regulate technologies, they regulate products. And they often, with newly emerging technologies, particularly refrain from setting a particular standard thoughtfully and on purpose, because they expect that the technology will improve and move on. And if they set a standard here, then everybody goes to that and they don't try to take it higher. And so I think that we would absolutely expect the FDA to be highly demanding. They are highly demanding. I think the EMA is actually not quite as mature in their thinking as the FDA, but they're getting there rapidly. And we know those guys talk as they should. And so I think that we should be continuing to push those standards. I worry a little bit. We say set a standard in a new area like this when we don't want to set a standard. We want to set an approach that continues to improve.
Maintain a dialogue that's continuing to push as we learn. And yeah, that makes sense.
Yeah, and I will say we have been impressed by how much the various agencies have been trying to educate themselves and understand the different technologies and really understand what should and shouldn't be done right now. They're not ignoring this, they're not putting it all on us. They're really putting in a real effort, which is great to see. So.
Great. I do want to build off of Sandy, one of your comments around if there was lack of specificity, we wouldn't know in the next 10, 20, maybe even 30 years, and link back to the comment before that a lot of the potential here is potentially treating pediatric indications and getting to things early. How do we resolve that, right? How do we resolve the fact that we might be treating children where we might not know for decades with regards to what some of the off-target effects might be, just because we can't even anticipate them at this point?
It's a really important question, and I think anyone that treats children with bone marrow transplants or chemotherapy also faces the same question. And that's why I think these treatments should be reserved at the moment for seriously ill children. and should require a body of evidence that may include some time in adults before you go to children. As I said, it's all about benefit risk. Every decision on a medicine is about benefit risk. What's the benefit versus what's the risk? And when I look at the children with MPS 1 and 2 and the sequelae and consequences of that disease, I'm confident that we've done enough both with unbiased ways of looking for off-target, but also simple safety pharmacology, as you would do with any medicine, looking at animals for some period of time to make sure that there's no consequences. But we do it with a sense of great responsibility, and it's the thing that keeps me up at night.
Albert?
Yeah. Well, just to comment on that a little bit further, and I think the other aspect, because of the precision of all of our technologies at the DNA level, sometimes animal models and getting safety pharmacology in an animal model is also a little bit of a challenge because we don't have perhaps the specificity in an animal model with our candidate we're going to take into humans. But as it relates to pediatrics and rare disease, some of these diseases are devastating. So sometimes there may not even be an adult population that you could test in, and really trying to balance out that risk benefit for that patient population. If you can have a child go from, normally 1 to 2 years and get them out to 10, 15, 20 years, to me that's a huge benefit within that patient population, assuming all of the safety that we can build to go into that patient population in a confident manner.
Because there's the argument that the adult you go into may only get risk and not get any benefit because their disease is already established. They're already in a wheelchair, for example. And so all they get is risk. And that's why we would push that one should go to the patient population that really needs it as early as possible.
So it's about picking the right indication so that we can justify the risk in pediatrics rather than pediatrics necessarily in and of itself being meaningfully different.
Right.
Yeah. Well, I mean, it's driven by the biology of the disease, right? I mean, if it's a patient population that does live and gets older, it may be that the disease is too far established and the pathology is intractable, and you still need to treat children or not. I think one thing about treating a genetic disease is that the lesion is there from the beginning. But I think there's also, and we're, I think, as a field, embarking on this very interesting. It's a scientific question that's deeply rooted in how do we understand safety, which is the following. We know we can make molecules where there are no detectable off-targets at not only the limited detection, but the limit at which stuff's happening to our DNA just sitting in this room. I mean, our DNA is not this static molecule. And so it's being, there's a reason we have DNA damage repair mechanisms, and it's not just because chemotherapy does damage and it has to be repaired. DNA is being mucked around with constantly. It's a little terrifying, frankly. And so these, there is some level of DNA breakage, repair, et cetera, that is just in normal human physiology. And so if we're down in that level of, we can't detect anything because we're in that background DNA noise, then what's a signal and what's noise of something that might be observed 5, 10, 15 years down the road? I certainly agree with Sandy that it's important to follow these patients long term. The FDA demands that, and those who work in gene therapy, same thing. I think, I don't know if the current standard is 7 years, 10 years, it's moving around a little bit. But yeah, of course, any patient will be monitored for very long term. That's critical. But how do we understand anything we might see? How do we discern signal from noise? It's not just a question for companies. I think it's a question for medicine, it's a question for regulators, certainly a question for patients as well. And one that I think there's this context of specificity that it does become theoretical. Because if you can't detect any off-targets at all, but you see something 5 or 10 years down the road, was it signal or noise?
That's a good point, and it's a very difficult construct. How do you even consent a patient for something that is a lifetime event, and with a risk that is not quite known, and if it exists is very small? Patients, all of us people, are very poor at understanding what risk means. If I told you you had a 1 in 10,000, would you do something? If it was a 1 in 100, would you do it? I'm not sure I understand what those things mean. So one of the things that I was talking to my chief medical officer yesterday about was starting a discussion about how we inform patients better about this. Because the current consent mechanism is technically correct, but it may not give them all the information that they need.
Yeah, and it highlights the importance of educating the patient community in general around what is genome editing.
And when you work with some of these rare diseases, the communities are very, very diligent in searching everything that's written on your concept and talking about it. And so we've made a real effort to partner with these patient communities, partly because by talking to them we can try and describe what we do in a way that is meaningful for them to allow them to make the right decisions.
What are their questions? When we're thinking about patients and what they're looking to understand, what are typically patient advocacy groups, for example, what are they looking to understand?
It's everything you would, everything from the tactical piece of what does it look like, what happens, what do I get? And then there's the questions about what's the long-term benefit. We had a couple of boys, young men, and maybe they must be about 20, 21, and both with haemophilia. And one of them had decided that as soon as this was available, this is what he wanted, and he wanted to change his life. And the other one said, I'm a haemophiliac, I've grown up with this disease, this is who I am. Yeah. And didn't want his disease taken away from him. He wanted to treat it and address it, but it was part of his life now. And it was only through listening to their view of their illness that we realized the importance of these interactions.
Mm-hmm. Yeah, it's actually a really, really interesting point how not patients, people with certain diseases or disorders view themselves and their identity around that. Mm-hmm. You hear this a lot in what we would call the disability community. But so think about folks who are deaf, and cochlear implants is probably one of the most clear examples where there's a real debate in the deaf community about are cochlear implants a good thing or not, and because
it changes who they
because
are.
Deaf culture and will people have sign language, et cetera, et cetera. So if you talk to people in that community, it's a serious because they also say, look, I'm not a deaf patient, I'm a whole complete person. And it's really important to understand that. People with we're working on a form of blindness, among other things, and you hear similar kinds of perspectives in this community. And so I think that it's actually something that is, and I've heard it's in other contexts as well, that you have to find, it's how Sandy said, the right way to engage not only with people who might be candidates for therapies we're working on, but their families and their communities. It is much more subtle than you might think.
I think it's a really important aspect to hear from patient populations what that unmet medical need is, because I think we read a lot about it in textbooks and we get it from a biochemical perspective. And I think we as a community now coming in, we have the tools to attack the disease at the cause as opposed to treating a symptom. And so when you go and meet with these patient populations, you can get a tremendous amount of insight into what that unmet medical need is. And I think we can then utilize that information to put into our target product profile, if you will, to start to build out what do we want our medicines to treat, not only attacking the cause, but for the patient and also a lot of times for the caregivers. So we're talking about pediatrics, the parents and families of these kids. You get a tremendous amount of information from them to help you direct how you want to drive your therapy.
Yeah, we found it to be incredibly valuable to bring the patient groups in, and it's not just understanding the unmet need, but it's inspiring to our team. It makes us look at the technical challenges differently. It makes us look at safety differently, and really focuses and I think inspires our group to really give it everything we have before we really commit to something. So I think it's absolutely essential.
So coming back a little bit to thinking about platform validation and building to addressing some of these unmet needs, what do you think are the next big milestones when we're thinking about what we're trying to demonstrate and trying to achieve, taking a forward-looking view?
I mean, I can speak to how we're developing our platform. We always structured it to essentially answer the different translational questions. One is, How do you engineer the molecule to be very efficient, very specific? How do you assess specificity and to develop specificity analytical methodologies? How do you control what you're delivering, what you're dosing? How do you deliver it? And then how do you, what we call directed editing, how do you achieve the kind of genetic repair you're seeking? Different kinds of mutations or genes require different kinds of repair, different genetic manipulation. And each one of these has a spectrum of degrees of difficulty, right? For delivery, given existing technologies and physiology and things like that, certain tissues are easier to reach than others. Probably the eye, the blood, and the bone marrow and the liver are the easiest ones. CNS is probably the hardest. So there's sort of a spectrum of degrees of difficulty. So one is to say in each of these spectra, how do you kind of march down it to, as you expand what you can do technically, you essentially are swooping in more diseases you might be able to treat. Certainly the, obviously what Albert and his team is working on, you can tell by the name, part of that is just to try to get more to some of the, not just being able to knock things out, but can you knock things in, if you will. And that's where you start to get overlap with gene therapy. It's often I draw on whiteboards. I sometimes try these two overlapping circles, and one of them is gene therapy, and the other is gene editing. And look, there's some diseases you can only approach with gene therapy right now. Some you can only approach with gene editing. Some you can approach with either one. You're going to do maybe a slightly different kind of genetic manipulation, but you can approach with either one. Does this circle of gene therapy expand? And if it does this expand into gene editing territory or into other diseases? And how does the gene editing circle expand? Because I do think the gene editing circle is expanding rapidly, partly as we better understand how to control DNA repair mechanisms, as we better understand ways to engineer not just the place of cut, but the architecture of the cut, how we can take advantage of that to achieve different kinds of repairs. And then I think with regard to delivery, there you're looking at more complementary technologies that you need to bring together with the editing molecules in whatever form. And I think there's going to be sort of tissue by tissue. Once you figure out a tissue, you can work through many different diseases there. But it's going to be a little bit more tissue by tissue in terms of delivery.
I think,
please.
I was just going to say, along those lines, so the platform that we're developing, we have 15 novel capsids that have never been in people. So part of that is trying to develop and understand there's two circles of gene therapy and gene editing, because we can deliver both, but it's characterizing that first milestone of how do our capsids go, what tissues do they go into, how do they distribute, can they get into cells that aren't necessarily dividing, such as the CNS, and does that open the door? Can you go in with the gene therapy to kind of approach where perhaps high expression is okay, but in other ones where you may need to regulate that very tightly, you may need to go with a more of an editing approach. And I think really building that, and then coming from the editing perspective, is just walking along those lines. Can we get into the cell type that we want to get into? Can we engineer the construct on the inside so that we have maximum on-target correction, minimum off-target correction within the cell type that we're treating that allows us to correct a phenotype? And so some of these phenotypes we may be going after that if we can correct 10% of the liver, let's get over that hurdle. Yeah. Then can we actually boost that so that now we can get into diseases where perhaps you have to correct 30, 40% of the liver before you see a clinical benefit, and working our way through each of those with the platform.
I would look at it slightly differently. We just had a strategy meeting with our board that discussed this specific issue. And the way we are talking about it now and thinking about it now is there's gene therapy, genome editing, where we drop a new gene into the liver, transcription control, where we control the gene wherever it is, and finally, the final goal has to be gene editing. And I think to imagine there's any other way but that arc is a very pharmaceutical model, that we give medicines to patients, so let's give them genes, that the gene will be in their liver and will be the gene therapy will give them a certain amount of an enzyme or a protein that we hope will be the right amount for that patient. It would be so much better if you could solve the problem rather than address the problem, which is what gene therapy does. So I think the final answer has to be to go and find out what it is that's causing the issue, wherever that is, and edit that gene and bring it to the wild type variant that would allow the patient's own control of transcription, and regulation and the whole ecosystem that is around that gene to work naturally, rather than hope that what I put in the liver is the right amount. So there's a quote from Uber that I keep using, that Uber's not here to replace taxis, Uber's here to replace cars. We're not here to replace gene therapy. We're here to replace medicines. That if we can truly do this well, and I think our editing, we have the advantage that it's a eukaryotic system, it works in cells, it works in humans, it should work, and it's just a matter of getting there. What really is the bit that is the next stage that you're looking for is all about delivery. And at the moment we put things into the liver because that's where we can put it. Some people go for the eye because it's an easier place to get to. At some point, either through a new capsid or lipid nanoparticles or another thing that we've yet to discover, we will work out ways to get to any cell in the body. And at that point, it won't be gene therapy we want to do, it will be gene editing or transcription control. And what we're just waiting for is that moment and that delivery technology. I have great faith that that's going to happen, because in the freezer at Point Richmond, we have the solution for cystic fibrosis, we have the solution for Duchenne muscular dystrophy, we have the solution for, because we can edit any gene and any nucleotide. And it's just the matter about waiting for the delivery that will get you there.
So what can we do to accelerate that, ability to actually unlock the potential of, what we've already done in terms of thinking about ability to deliver?
Meetings like this brings people together that are in delivery companies and in editing companies, and we talk to each other, and we even have a beer with each other, and we stimulate that desire to deliver now. And as well as doing our genome editing, we spend a lot of time looking at the adjacencies of delivery and do some work on it ourselves, but also talk to the people who are doing it. And I'm sure all the people here watch the delivery field with great interest.
I think it is, though, an area where I would not anticipate there's one technology that unlocks lots of tissues. I do think it's going to be much more tissue by tissue. I mean, certainly you see in, for example, the world of AAV, there's a lot of folks working on variants to try to, whether they're discovering naturally occurring variants or doing engineered variants to try to modify the properties to get into other tissues. But even there, I think where we see the most intriguing work is where somebody approaches it with a deep understanding of a particular tissue or particular biology, and so can really bring to bear that understanding of the physiology and with a biological goal in mind. And so, there's, talk about all the different technologies out there, but I think even, lipid nanoparticles is a phrase that often gets used, and certainly some folks in this field are using that. But there are 1,000 lipid nanoparticle technologies. It's not one technology. And really getting it and fine-tuning it to work in a particular tissue requires effort and focus on that tissue, the physiology of that tissue. So I think that the journey, as a practical matter, will be a long one, in that it's not, I don't think there'll be one solution that opens up all the tissues of the body. I think it will be a bit more of a, beachhead by beachhead.
One of the other things that we've been thinking about as well is, can you get to a point where you can multi-dose? And so can you get some delivery initially? Can you come back in with a little bit more delivery? Can you come with a little bit more delivery? And really working on that, trying to trick the immune system into allowing you to do that so the cells will take that up. And I think that, coupled with, there's a tremendous amount of work in the device field. So if you know what cell type you really want to go into, it is a one-time therapy for a lot of what we're doing. Can you use that delivery device to put it right where you want it, and sort of marrying those two? So there's the biological mechanisms of delivery, but then there are a lot of device advancements that are being worked on as well. So bringing those two fields together, I think, can help.
Yeah, and to go back to what we talked about earlier, I think this is why it's so critical that we have a few early wins, a few clear signs of human proof of concept that will, even if it is directing to the liver, the eye, the blood, that's going to drive other groups to work even harder to solve what we call the delivery problem in the field, to really open things up. If we don't have those early successes, it's not going to drive those groups to push as hard and as fast. So today, while maybe frustrating at times to say, well, we've got to leave the cystic fibrosis program in the freezer. By focusing on the programs that we think have lower risk from a delivery perspective, I think that is the most prudent path for us to take right now, because long term, I think that is what's going to open the door to these other areas.
And I mean, if you want to, we all have a great example in front of us right now with Alnylam, right? So 17 years old, finally positive phase 3 data on a first program. They're focused on the liver because that's the tissue they were able to solve, and there's tremendous opportunity there. When they were founded, and actually is on my board, John Marigner is a good friend. I mean, I think it's amazing what they've done. But I think when they started, they saw the possibility of RNA as a totally different way to drug biology of any target. And it is still incredibly powerful, but how broadly will they be able to make medicines? Right now they're really focused on the liver, and I don't know to what degree they are going to start to invest in other things. They've always had some, if you they present on their data in other tissues. But when does that turn into a development pipeline, for example, that then stands up alongside the liver? I think that's not tomorrow, I don't think. And so I think it just gives you a sense of, for a company that really pioneered a brand new modality, what does that journey look like?
And it's a journey. Yeah.
And in their case, $1.8 billion to get to this point. And a lot of things that didn't work along the way, because that is what you have to do. They obviously also invented a delivery technology. They didn't get it from the outside. They did tons of early collaborations with device companies and different technology companies. And then GalNAc ultimately came from their own labs, which is also kind of interesting. I think we all probably face this tension of how much do you think you might find the technology from the outside, and how much do you think you understand the problem better than anybody, so maybe you need to solve it yourself. And we can have an existential debate about that one. But I think that we can all learn lessons from other places in the industry, and I think Alnylam's a pretty interesting one to think through. What does it look like to pioneer a new modality, and how do you think about investment in your platform on an ongoing basis? Because GalNAc was not the first thing that they did. Right? They had other approaches that turned out not to work, and this was the one that worked. And that came because they invested in their platform. And for them, that was, once they solved the basic, some early stuff about RNA chemistry that they had to figure out, that they worked that out over the first 2-3 years. But then the long-standing platform question for them was delivery.
Great. So we only have a few minutes left. I think we've heard Stanley's point of view with regards to what is the potential and what is the future and what are we building towards. I do want to open it to a few of the others, maybe Matt. What is your hope, and where do you think that we'll be in 15 to 20 years as we start to think about the potential of genome editing?
It's interesting. I'm looking at our names of our companies in front of me here on the slide. One thing you might notice that's different about ours is it doesn't say medicine, it doesn't say therapeutics at the end of our name, and there's a reason for that. We actually see, and I'll upset everybody at the cell and gene therapy meeting here, we actually see the greatest near-term potential for genome editing, not in, call it classical human therapeutics or human diseases, but in food, in plant agriculture. We think that's where we can actually make the biggest global impact near-term. So within that 10 to 15 year horizon, given the different environmental and social constraints that are being placed on our food system right now, that's where we think 10 years from now people will be talking about editing the most.
Matt, how do you feel about the societal element there, and what can we learn as we go into humans about that debate as happened in agricultural science?
Yeah, no, it's a great question. We certainly don't want this field to end up with the stigma that say the Monsantos of the world have gotten with GMOs, and so I think it is going to continue to be, and we have been very focused on engaging with all of the different food industry groups to make sure that what we are doing, the approach that we're taking, is being clearly explained, very openly explained. One thing that we kind of learned from the lessons of the GMO guys from the early 90s was, don't just put it in the market and then let the public find out about it later. We want to be upfront about what we're doing right from the beginning, explain the benefits, explain the risks, and then have that open dialogue. And so far, we've been really encouraged by what all of the different food groups and various groups that normally you would think would be against a biotechnology approach, and really encouraged by their the positivity that they're taking towards editing. They realize what the challenges are today. They're actually far more educated than I would have thought they would be from a deep technical level. And I think the future is bright for editing in the globally within the food space.
So I see just 15, 20 years, and where I see this big influx coming, we have the Human Genome Project. And then you had that aligned with a lot of human genetic studies to understand what are the monogenic causes of a lot of diseases that 20 years ago we did not know. And so now we know, the cystic fibrosis, the Huntington's, a lot of inborn errors in metabolism. We know what genetically causes those. We then have newborn screening lined up. So I see 10 or 15, 20 years from now, the ability to go in and correct those, capture those at newborn screening, understand it in that patient population, and fix it. I think we have to walk, because the biology as well around these monogenic disorders, it's one gene that has a very large effect size. Beyond 20 years, we may get to a point where we now understand more complex diseases with multi-gene, that we can start to go and attack that as well. So that's kind of where I see the whole field going, is leveraging what we know about the genetics and the biology and the physiology of these diseases, and pointing us to where we can go in and make those cures.
Great. Katrina? We'll solve the technical challenges. I mean, some sooner than others, but I think that the idea of gene editing is more an engineering problem than a science problem at this point. And when you get to that point, then it's the biology that becomes rate limiting. If you look at one of the other areas I kind of think of as an interesting comparator is antibodies, right? And if you know when the first antibody was approved, 1986, it was fully murine. And it was nearly a decade before the second antibody was approved. And there were two same year. One was CDI grafted and one was chimeric, right? Now, clearly, a murine antibody is not very medicinal, although OKT3 is still out there. It's still a medicine. But you had this desert of a decade. And what was going on? Not only figuring out how to humanize, but also some of the less glamorous stuff, the engineering problems of how do you make it, how do you get a high concentration solution, which frankly, if you want to make a medicine, you're not going to get a publication in Nature Science for doing that, but you actually have to go through that gauntlet to get to medicines. So we had a decade where all of these problems were being worked out. And then somewhere in that, late 90s, if you plot how many patients and how many different antibody drugs are treating how many different patients as like a stacked bar graph, I know this because I built this at one point. It does take off like a hockey stick. And part of what's fascinating is not just the number of patients treated, but the diversity of targets and the diversity of diseases and the diversity of biologies. So in that same time frame, you have people understanding biology and knowing where to then apply this tool that's finally been honed. And so if we were to say, what's the gene editing equivalent of that graph? Are we at OKT3, or are we actually 5-10 years before that? Or are we at the point where the graph is going to break out? I'm not sure I know that. I don't have a crystal ball that's that good. But I do know that as we solve the engineering problems, biology is really what becomes rate limiting. I do think, Albert, your point is an excellent one, which is the genetic foundation that we've built through the Human Genome Project and everything since then becomes critical. And if you look at, many of you may have seen this plot of, it's actually the y-axis is a log scale, and the x-axis is time. And if you look at the cost per base pair of sequencing, it comes down at a pretty steady rate that essentially tracks with what Moore's law would propose. Moore's law would say it keeps going like this. What actually happened is it goes like that, fell off a cliff, which is fantastic. More important, I would argue, is that in the same time frame that you saw the cost drop, which you could argue for $1,000 genome or not, it's somewhere around there. At the same time, accuracy and speed increased greatly, because to get to this being a real tool for physicians, for decision making, et cetera, it can't be that it's 6 months to get a sequence. It can't be that, yeah, it's pretty accurate. Yeah, it's pretty good. If you're looking for a single base pair mutation, so the science of sequencing, has matured tremendously. Not done, but matured tremendously. So we have this background of genetic infrastructure, if you will, both technologies and knowledge, that is also, now you see all these different initiatives, a million genome database here, a million genome database there, in particular where it's properly consented and connected to healthcare records. So you know what happens with that gene, that genetic formula in life. And those sorts of they're really just, it's an investment in knowledge bases, will become the raw material for the biology. Because, as you also pointed out, animal models, they have some inherent limitations. If we can start with the human biology, and we're getting to that point not only of knowledge of the genome itself and that sequence and the tools to know what yours is and mine is, but in the context of healthcare information, again, properly consented, that really does allow the hypothesis to start with human biology, that puts us, I think, in a much more powerful place to envision what kinds of medicines we can make.
Great. So there's a couple minutes left. Are there any questions from the audience? Okay. Well, thank you so much for your time.
Thank you.
Thank you.